Okay, we are going to evaluate some competencies that you have to explain the functioning of chemical and biological systems through structure and coherent arguments based on concepts, theories, and principles of natural sciences, mathematics and computing, and the scientific thinking. Okay.
Okay.
Okay. You have to do the research about human papillomavirus. And what can you tell me about the initial, middle, and final stages of this HPV?
Mm, well, as we know, HPV, someone can contract HPV for a sexual interaction, but the virus can just have access if there are any abrasion or cut in the epithelial cells. So if we have some of these affections, the virus can enter and infect the basal cells. After this, the um, virus produce E1 and E2 proteins that at the same time are following by the famous oncom proteins E6 and E7. These proteins, E6 and E7, are responsible for this regulate the replication of the epithelial cells, and then uh, the viral infection generates L1 and L2 genes. That means that have that can infect and all the basal cells progressing into a cancer.
Okay, thank you. Okay. If you want to detect the presence of HPV in the patient, which molecular technique will you choose and why?
For detect an infection of HPV, there are many techniques, but the most common and efficiently and specifically with the results is PCR, because PCR um, uh, con- extrain all the viral DNA and separate them in small fragments. And if we can do this, we can detect an early infection of HPV. And PCR works uh, with a fluorescent, no, with electrophoresis in gel. That basically you collocate the viral DNA in agarose gel, and then with an electric current separates in smaller fragments the viral DNA, and obviously the smaller fragments travel farther. But if the band of the DNA extract is similar with a size is expected, that means that if we have an infection.
Okay. And what advantages do you find in PCR and what disadvantages?
Well, one advantages are maybe it's a very common technique for women is very um, clearly with the results. But one the disadvantages of this technique is that, for example, don't detect say if the patient have an active infection. Just detect the infection of the virus, not an active infection.
Okay. And do you know any hybridization molecular technique?
Yes.
How do they work?
Uh-huh. I know two. One is hybrid capture, and the other one is DNA hybridization arrays, or known as microarrays. So for microarrays is a very excellent technique because not just detect the HPV infection, also detects all the genotyping of high risk HPV. So for this technique, you extract the viral DNA, and you will label it in a fluorescent tag. Then the DNA is hybridized in a glass slide, and if we have a positive result, is because the DNA, well, the viral DNA strand are similar with the size we are expected. And for hybrid capture, we extract the cervical sample with a special brush. After we extract the, the sample, is uh, is mm, the DNA is mixed with a cocktail of RNA, high risk HPV. After this, with antibodies, you can capture all the mix of RNA and DNA. Also, to interpret the results for for the two techniques, we use a fluorescent dye to visualize the results in a UV light. And for hybrid capture, depend on the intensity of the light signal is the type of the infection of HPV.
Okay, thank you. Mm, do you know which are the cancer biomarkers related to HPV?
The key biomarkers related to HPV are the oncoproteins E6 and E7. As I mentioned, these proteins are responsible for disrupting the replication of the basal cells. But mm, to detect these proteins, we use a technique called the dual strain or P16/Ki67 test. For this test, we extract the, the sample and uh, how we extract the sample. And uh, it's similar like the other techniques like the fluorescent dye.
Mhm.
And if we have P16, means that we have a presence of the E7 viral proteins. That means that we have an active that the viral genes begin transcript effectively, while if we have in the results Ki67 means that we have an abnormal and uncontrolled cellular division with the viral genes also. And if we have these two makers in the same cell, that means that we have an active infection. And that's it.
Okay, thank you. And the last question. How do you think Mexican population can benefit from molecular techniques?
Well, I think that all women in the world can be beneficiated with these techniques because uh, most of the people, and especially women, think of believe that this infection or this disease don't exist or is like mm, not important. But uh, I think that all women can consolidate a very good life, a better life, not just for themselves, not all the family, all friends. Maybe these techniques are expensive, so not all the people can have access to them. But maybe no, well, no, maybe. I think will be a very mm, very excellent for women because for an example in rural zones you know that most of the women have children in a very early age and maybe in some cases can contract the infection and pass the time and don't check out the body the health and obviously die but maybe this will be help all women but obviously it's important to inform not just women also men because I know that men are are the infect persons. The men have the virus, and women are responsible for developing the virus. So I think that would be a very uh, important, special help to all women.
Okay, thank you.
Thank you.